Hello. Welcome to the next in the series of review clips designed to assist you with preparation for your Biology 3201 public examination. In this clip, we'll be examining some variations that occur on the Mendellian theme. Specifically, we'll be taking a look at incomplete dominance, codominance, and polygenic inheritance. Mendel was fortunate that when he selected his traits, he was able to gather a simple system. Traits that simply had yes or no. The one caution I will give is that don't confuse dominance with abundance. And dominance relationships vary on a continuum from complete dominance, as was evident in Mendel's traits, to incomplete dominance, to codominance. Incomplete dominance, you get a third phenotype, an intermediate, whereas codominance, both alleles get expressed at the same time. With incomplete dominance, the heterozygous condition displays an intermediate phenotype. It's usually uniform, and if you didn't know about the homozygous phenotypes, you might not know that there were two different alleles at work. Whereas codominance, the phenotype that's displayed expresses both distinctly. Generally, it's not uniform, and traits from both parents can be seen. Traditionally, under Mendel's experimentation, if you have parent 1 which is white and parent 2 which is dark blue, in the offspring you would see if blue is dominant, all the F1 would be blue. If white was dominant, you would see all the F1 generation being white. With incomplete dominance, what you would see is a third or intermediate phenotype. White and dark blue gives you a completely uniform light blue. Codominance, on the other hand, you would see the following. Both are being expressed. There are portions of the offspring which are blue, portions, of course, which are white. This is codominance. Incomplete dominance, uniform, a third phenotype. Codominance, both traits being expressed. Again, parent 1, white, parent 2, dark blue. Incomplete dominance gives you a third phenotype, light blue. Codominance, both are being expressed at the same time with blue and white sections. Incomplete dominance, codominance. But the reality is that once you get down to the molecular level, even with incomplete dominance, what we see is actually codominance on a much smaller scale. In flowers, this commonly occurs. For example, in snapdragons, red and white. The F1 generation will give you pink. The heterozygous condition reveals a pink phenotype. And when you cross the F2, you would get 1/4 red, 1/4 white, but you would get half of them being pink or heterozygous. With complete dominance, the dominant allele creates the full phenotype of one of two methods. Half the amount of gene is being expressed, or it's upregulated to match the homozygote. Whereas in incomplete dominance, the recessive allele is not expressed in the heterozygote. The homozygote gets 2 doses, the heterozygote gets a single. And in codominance, both get expressed, resulting in a combined phenotype. Another variation on the Mendellian theme is that of polygenic traits. Mendel chose traits that were simply yes or no. There was very little by way of range. Oftentimes, any trait that shows a range, height for example, weight, skin color, eye color, are usually traits that are affected by alleles at many different genes. Polygenic traits are actually common. Simple ones, simple one locus, one location, such as Mendel studied, are actually relatively rare. Polygenic traits typically show a continuous variation, that is, a bell curve, and relatively unpredictable inheritance. A model you can see here. For example, if you had a trait which is composed by many genes, polygenes, in this case A's, B's, and C's, depending on the extent of A's, B's, and C's dominant recessive, that determines where it falls on the continuum. For example, little a, little b, little b, little c would be on this end of the spectrum. Big A, big B, C would fall on this end. In between the two extremes would fall all of the different ranges, depending on the combinations. The completely heterozygous condition would fall right here in the middle. Eye color in humans is a common example of this. Eye color being governed by two genes, A's and B's. And depending on where it fits, that determines the eye shading and all the different eye colors that correspond with it, as you can see in the sample Punnett square. In past public examinations, there have been questions on this topic. For example, what type of inheritance causes variation in height and skin color in humans? Codominance deals with both being expressed at the same time. Incomplete dominance involves a third intermediate phenotype. Multiple alleles would deal with blood types, which we'll look at in a later MLO. The answer to this question, the correct answer, is D, polygenic inheritance. This question is posed. Ask what inheritance pattern controls trait that shows continuous variations, such as height in humans. For the same logic, polygenic inheritance would be the correct answer. This question asks, a white bull is crossed with a red cow. All the offspring produced are roan, containing this key, red and white. Both are being expressed. The correct answer for this one would be A, codominance. Incomplete dominance would result in a third phenotype, not both being expressed at the same time. Sex-linked inheritance involves transmission which is linked to one or the other of the sexes. And polygenic inheritance involves multiple alleles looking at a range. The correct answer is A, codominance. Constructive response questions have also been addressed as it relates to this topic. This question asks, you're told that you have a table below that shows the genes involved in eye color. You're given the genotype with this corresponding eye color that would result. You're asked to use a Punnett square to determine which eye color is possible for children born to the couple. You're told the man has gray-blue eyes. Gray-blue, so his genotype would be as follows. The woman has green eyes. Therefore, her phenotype with green eyes is as indicated in the table. So the parental cross that we have is, for the man, big A, little b, little b. For the female, She is little a, big B, big B. You're asked to complete, and you are told to use a Punnett square. To do this, the first step is to look at the possible alleles. Again, this is the man, and as a term from the chart, the female is as follows. The possible alleles, gametes being created, are big A, little b, little a, little b. Again, using the FOIL method, I can determine this by pairing up all my possible combinations. There are only two possible alleles from the man. From the female, using the same methodology, all of her gametes will be the same, little a, big B. Completing a Punnett square for this is a matter of actually just writing in the possibilities. The female, remember, only has one possible gamete, so I can do an abbreviated Punnett square. Her gametes are going to be little a, big B. For the male, the possible gametes are as follows. Big A, little b, little a, little b. Therefore, the only possible gamete combinations, genetic combinations in the offspring, are big A, big B, little b, or little a, big B, big B. Referring back to the chart as presented, you can very easily see what the possibilities are. Remember, you produce big A, big B, little b. Big A, big B, little b gives you the following. Light brown. The other individual, the other possibility, is little a, big B, big B. Little a, big B, big B would give you green. The question asks, what possible phenotypes could be created? The answer then would be light brown and green. And again, my suggestion to you is that at the end of each question, write your answers very clearly. Make a statement. Possible phenotypes equals light brown and green. Even draw a square on to clearly draw the attention of the markers to your answer. Thank you very much. I hope you found this learning clip to be helpful.